Welcome to Biology Practical of the year 2013. I will first of all take you through the questions, and after I take you through the questions, I will now give an elaborate way of how the practical was marked. So, welcome to 2013 Biology Practical Marking. So, we start with the questions. Question 1. The paper was taking 1 3/4 of an hour. The photograph shows the inner surface of the upper left side of the rib cage. And you can see it there with the labels K. You have also labeled rib and label M. Question 1. Name the bone covered by the fatty tissue labeled K. That is A1. A2. Explain the role of the part labeled M in inhalation. Question 1. There is another photograph below which shows a mammalian vertebrae labeled S, T, V, and neural arch. Question 1 still continues. In this one, you state the view of the vertebrae. And 2, name and state one function of the part labeled T. Name and function. B. How are the parts labeled S and V adapted to their functions? C. The actual width of the vertebrae below in centimeters is shown by a section of the ruler in the photograph. So you can see from A to B. And you can see the actual length which you are supposed to read. The question is, C1, determine the width of the vertebrae on the photograph. Calculate the magnification of the image and determine the actual length of the vertebrae from point A to point B, showing you are working. Question 2, you are provided with food sample labeled solution C. Using the reagents, carry out a test to identify the food substances present in the sample. So that was your table, reducing sugars, non-reducing sugars, and proteins. That is test 4, procedure, observation, and conclusion. Question 3, below were photographs showing some observable feature of leaves. So you have compositae, papilionaceae, comedinaceae, malvaceae, bignoniaceae, and nystaginaceae. I don't know whether those names are quite visible. Let me see. Yeah, I think I have to produce that one again so that you can see the names. So I've repeated with a bit magnified photograph. So you can now see very clearly compositae, papilionaceae, malvaceae, bignoniaceae, nystaginaceae, and comedinaceae. Using the features in the order given below, construct a dichotomous key that can be used to identify the specimens. You get 10 marks. So there is simple or compound leaves, leaf venation, leaf margin, arrangement of leaves on the stem, pinnate or trifurate nature of ribs were the steps you are going to use to make a dichotomous key. Remember, for each of the features there, you have two alternatives. All right, dichotomous, two alternatives. And di means two statements in each of those features. Name the bone covered by the fatty tissue level decay. I'm now giving you the answers. The bone is known as sternum. Accept breastbone as one word. Reject spelling mistakes. Reject breast bone with a space or breast hyphen bone. The role of part M in inhalation. M is internal intercostal muscles. So the internal intercostal muscles relax. Or M, or it relaxes, or relaxes, pulling, or causing, or allowing. Remember, guys, you select one. The rib, or the rib cage, to move upwards and outwards. This increases the volume of the rib cage while the pressure decreases, forcing air into the lungs. You get 6 marks, and the maximum you give there is 5. Note, mark the identity first, but the rest is marked when it appears. To score a mark in volume, increase and decrease must be mentioned. Reject everything if M or external intercostal muscles contracts. You have to start with the relax, because this is inhalation. State the view of the vertebra presented. This is anterior view, or you can say dorsal view. Name and state one function of the part labeled T, and the name is neurocanal. Reject wrong spelling or front view. Function, passage of spinal cord. But you reject it contains the spinal cord as a function, or passage of blood vessels and spinal cord as a function. The function is passage of spinal cord. Determine the actual length of the vertebrae from A to B. Show you are working. S is neurospine. It is wrong to provide a large surface area for attachment of thoracic muscles. Centrum V, it is thick, or solid, or broad, or wide, or large, for bearing or for supporting the weight of the body of vertebral column or other vertebrae. Please know how to select the ones there. Reject any specific body parts, all right? And weight of vertebra decreases downwards. The actual width of the vertebrae in centimeters is shown by the section of the ruler in the photograph. And when you read, it is 9.8 plus or minus 0.1 centimeters, which can give you 9.7 or 9.8 or which is this? Let me see. There was an error. It is 9.8 plus or minus 0.1, which now corresponds to 9.7 or 9.8 or 9.9. Before it had been written as 0.0, but it is 9.9. Accept without units, but if used, should be correct ones. So you don't need to write the centimeters, but it is advisable as a scientist to write the units. Magnification is image width over actual width. So 9.8, and width is 4.64, all the way to 4.7. Then you get the magnification ranges from 2 to 2.13 times. So all these answers you see here are possible answers. But you can accept rounding off 2 times 2. You can accept it. So all those workings there are because you have alternatives, like you work for 9.7, you work for 9.8, you work for 9.9, and those are the answers you're supposed to get. And round off maybe to 2. Determine the actual length of the vertebra from point A to B, show your working. So image width over magnification equals 10.4 plus or minus 0.1. We are allowance, that's the allowance. You divide by the magnification, and you are going to get 4.84 centimeters, lowest actual length. All right. So you can have now 10.5, you work. Okay. And that's what you are getting, 5.25 centimeters as your highest actual length. Then you work the others, because you can also have 10.3. All right. Get the answers. You can also get 10.5, and you work everything. But all those ones will be equivalent marks. Determine the actual length of the vertebra from point A to B, show you are working. So there you are. C1, C2, and C3 are tied. So if you fail C1, you cannot get C2 and C3. Question 2. Test, procedure, observation, conclusion. I don't know, my guys, whether you will be able to read, you are able to read this, but you can be able to read, guys. We have reducing sugars. Put 2 cubic centimeters of C in a test tube and add equal volume of Benedict solution. Or you can say D and put in hot water bath, or heat, or warm, or boil. No color change. Blue color remains, or color of Benedict solution remains. Reject, no change. No observable change, no reaction. You have to say no color change. Then you conclude that reducing sugars are absent. Non-reducing sugar, chemical formula must be conform, or must conform to UPAC rules. All right. Put 2 cubic centimeters of C in a test tube and add a few drops of diluted hydrochloric acid. Place the test tube in hot water bath for hot water bath for 3 minutes. Remove the test tube and cool in cold water. Add sodium hydrogen carbonate, drop until fizzing stops. Add 2 cubic centimeters of Benedict solution. Place the test tube in hot water bath, or warm, or boil. Color changes to green, yellow, orange, brown. Sequence must be correct. Color changes to green. Reject color changes to orange or to any other color. Non-reducing sugars present. Or if you say the color changes to green, you can conclude that there are traces of or retro non-reducing sugars. Reject reducing sugars present. Proteins. Put 2 cubic centimeters in a test tube and equal amount of sodium hydroxide solution. Color changes to purple or violet or mauve. Accept colorless to purple or violet or mauve. Protein present. So this is food tests. Question 3. These are the answers. And we have the first type or the first alternative, where we are having simple leaves, compound leaves. For simple leaves, you can go to 2. For compound leaves, you can go to 4. Leaves net veined, reticulate or branched, go to 3. Leaves spiral veined, you have identified it is Commelinaceae. Leaves with serrated margins, that is 3A, Malvaceae, you have already identified that one. B, leaves with smooth margin, Nystaginaceae, you have identified. Leaves opposite, go to 5. But leaves alternate is Bignoniaceae, identified. Leaves pinnate is Papilionaceae, and leaves trifoliate is Compositae. So you are going to 2 and going to 4, go to 3 and go to 5. So that is the dichotomous key. So another key would look like this. Leaves compound, go to 4. Leaves simple, go to 2. Leaves net veined, go to 3. Leaves spiral veined, Commelinaceae. Leaves serrated, Malvaceae. Leaves with smooth margin, Nystaginaceae. Leaves opposite, go to 5. Leaves alternate, Bignoniaceae. Leaves pinnate, Papilionaceae. And leaves trifoliate, Compositae. And finally, you have the third one. Leaves compound, go to 4. Leaves simple, go to 2. Leaves spiral veined, Commelinaceae. Leaves net veined, go to 3. Leaves with smooth margin, Nystaginaceae. Leaves serrated, Malvaceae. Leaves alternate, Bignoniaceae. Leaves opposite, go to 5. And the first feature, leaves trifoliate, Compositae, and leaves pinnate, Papilionaceae. So any of those 3 dichotomous keys could have been produced by the students. So that brings us to the end of the way the practical of 2013 was marked. Guys, wait for the 2014, 2015, and 2016 coming soon. Don't forget to hit that button, subscribe to this channel, give a comment for any improvements you want me to make in the other upcoming videos on biology practical. Goodbye guys, and see you in my next video.